Welcome back to my channel. This time we are going to be having a class on genetics and variation in the world of biology. So this video will be of help to those students that are preparing to writing exam. Alright, so let's dive into looking at it. I'll be stating the main things that you need to take note of about genetics and variation. Now this is like two topics in one, but it's one that is giving birth to the other. What do I mean by that? Now, looking at genetics, genetics is simply the study of heredity, heredity and variation. The study of heredity and variation. That's what genetics means. And that word heredity is inheritance. Inheritance. All right. So what are transmitted from parents to offspring is actually inheritance. What do offsprings inherit from their parents is under this a part of biology called genetics and variation. While variation talks about marked differences amongst the organisms present in a given species or amongst a population of a given species, the differences that you can find, because not every individual of a given species are actually alike in every aspect. You get, in terms of their physical structures, in terms of their functionalities, they are different. So we are going to focus on genetics first before moving on to looking at the key things about variation. Now, considering genetics, the word genetics was coined by a scientist named William William Bateson. William Bateson. So William Bateson was the scientist that coined the word genetics. And one that studies genetics or majors on the study of genetics is known as a geneticist. A geneticist. All right. So let's see some important terms that are associated with the word genetics. Terms like we have traits. Now, traits, they are also called characters, or you can refer to them as characteristics. All right. Then we have the alleles. Alleles. We have gene, we have genotype, we have phenotype. So these are some things that are associated or terms associated with genetics, singular locus. So let's treat on these terms one after the other quickly. Now, considering traits or characters or characteristics, we are simply referring to the genetic as well as physiological attributes of an organism. All right. And considering traits, they could be of two kinds. Now we have dominant, and we have recessive. These two terms, you shouldn't be confused about them. Now, dominant trait or dominant character simply means a character that is expressive in the presence of another suppressed character. You get the point now? So the one that expresses itself is known as dominant. Then while the one that is hidden or that is suppressed is what? Recessive. Now, for dominant, dominant can be expressive in both conditions. What do I mean by both conditions? In homozygous conditions as well as heterozygous conditions. I would explain that. What I meant by homozygous conditions and heterozygous conditions simply means that homo means the same, hetero means different. So when you have a pair of same character, it's called homozygous. Then when you have a pair of contrasting or different characters, then it is heterozygous. What do I mean by that? Now, for example, let's say I have T, T, and T, T. If you observe, these are big letter T's, while these ones, they are small letter T's. Now the big letter T there means tallness. Then the small letter t means shortness or dwarfness. So looking at these letters, they are like the genes, all right? So if you consider this is in an homozygous condition, so you can call this homozygous tallness, while this one is homozygous shortness, because we're having same letters written. What about when you now have different letters? For example, big T, small t. In this case, this is heterozygous trait of tallness. Now, this big T is dominant over this small t, meaning that tallness is always dominant over shortness. I can actually explain this using colors. For example, let's say we are having a container containing black color and another container containing white color, and then we have an empty container, and then we are told to mix these two colors. So if you turn white and then you turn black and you mix them, the one that would be expressive, of course, should be the black color. All right, so that black that surfaces, that reveals itself, is actually the dominant one because it is expressed, while the other white will be suppressed. All right, so that white is considered as recessive. So I can say that dominant is always expressive in both conditions, whether homozygous or heterozygous, meaning in this case, let's work with this now, then you understand better. Now the tallness here is dominant, as well as the tallness is still dominant here. So what you can see phenotypically in this case is that the person is tall, and this one is also what? Tall. So the tallness is dominant in both conditions, whether homozygous or heterozygous. But looking at this one, for recessive, recessive cannot be dominant, or recessive cannot be expressive in heterozygous conditions. What do I mean? This one now. The recessive, which is the shortness, is not being expressive in this heterozygous condition. It can only be expressive in what? In homozygous condition, such as something like this. So in this case now, this is homozygous shortness. So it can only be expressive in homozygous conditions. So don't be confused. Just know that recessive is only expressive in what? In homozygous conditions, but dominant can be expressive in both homozygous as well as heterozygous conditions. All right. So that's what this thing entails. Then next is alleles. When we talk about alleles, we are referring to different variations of the same gene. Different variations of the same gene. What does that mean? Now you could have a gene coding for, let's say, brown color eye, or a gene coding for color of eyes. Let me use it that way. A gene coding for color of eyes. Now that gene could be coding for brown, it could be coding for blue. So these two colors now, they are different. So I can as well say that that is alleles. So I can say alleles for brown eyes, alleles for blue eyes. So that's how it works. So it's just referring to different variations of same gene. Then next is gene. What does gene represent? Now, gene is simply the unit of inheritance. A unit of inheritance. When I say unit of inheritance, it means the foundation of inheritance is gene. It's just like you saying the unit of life is cell. That's how you say the unit of inheritance is what? Is gene. All right. If you take a good look at the cell, let's say an animal cell. Now you have you have the nucleus in the cell, and then you have thread-like structures that are called chromosomes. And then within the chromosomes, you now have the genes. And within the genes, then you now have the DNA. All right. So we have chromosome, the chromosomes. So the chromosomes within, you now have the gene, and within the gene, you have the what? The DNA. All right. Now, these chromosomes, they are like thread-like structures that tend to carry genetic materials. That's the reason why if you must have read about the cell, they will tell you that the part of the cell that carries genetic materials are the nucleus, or the nuclei rather. So the nucleus is basically involved in carrying what? Genetic materials. And as a matter of fact, in man, we have 46 chromosomes. And these chromosomes, they are shared into two parts. We have 23 from father and 23 from mother. All right. And then in this 23, some are actually sex chromosomes. I don't want to go into unnecessary details, but of course, you ought to know that we have the X and the Y chromosomes. They are the sex chromosomes. Now, the X chromosome and the Y chromosomes basically are sex chromosomes. And when you have the X gametic of the sperm fertilizing the X of egg, it forms what? A female child. But when you have the Y of sperm, because in sperm we are having both the X and the Y gametic. So when the Y fertilizes the X of egg, it gives rise to what? A male child. So that's under fertilization. Then coming back, now the DNA is also known as deoxyribonucleic acid. Deoxyribonucleic acid. All right. So that's the full meaning of DNA, deoxyribonucleic acid. Now, we have seen that the gene is the unit of inheritance, and within the gene, you are having the DNA. Now, the location of gene in chromosome is what is called this loci, plural, then locus, singular. All right. So take note of that. Then the next is genotype. I'm still going to revisit the DNA for you to understand something about that guy. But let's quickly look at genotype and phenotype. Now, what is genotype? Genotype refers to the genetic makeup. This is the genetic makeup of an organism. Genetic makeup of an organism. All right. So it refers to the sum total of all the genes an organism inherits from both parents, talking about the father as well as the mother. All right. So when we talk about genotype, most times students they often refer to genotype as the AA, the AS, the SS, we have AC, we have SC. Of course, these are the common ones. AA, that's talking about the genotype of blood, then AS, then SS, AC, and SC. But the one that is common is AA, AS, and SS. Now, this one is known to be normal, then this one is known to be carrier, then while this one is known to be sickler. All right. But the best genotype is actually AS, the one that is even known as the carrier. The reason is because, according to World Health Organization, they recognize a particular illness known as malaria. And this malaria can result in death if not controlled. All right, death of population. So the best genotype that has the strongest resistance to malaria attack is AS. All right, so that's for genotype. Then for phenotype, phenotype and genotype shouldn't be mistaken for one another or each other. Now, phenotype refers to the physical appearance or attribute of an organism. Like, what can you see physically about this organism? Of course, it can refer to color. Now, you know there are some persons that are dark complexioned, while there are some persons that are light complexioned. That's in terms of color. Then, as well as height, yeah, some are short, some are tall. In terms of size, some are fat, some are slim. So those are things that you can see. But looking at genotype, you cannot see genotype. You can't look at somebody and then tell the person that you are AA. It's never possible because it's resided within the gene. So it's not being expressive. You can't see a person and just guess the genotype, You get. So for phenotype, is physical attribute, physical appearance. All right. That's for phenotype. Then let me just quickly run through this DNA. Now, considering DNA, the DNA is a double-stranded helix. What do I mean? Now, when I say double-stranded, it's twisted. The DNA is twisted. Now, looking at the DNA, you know that you have strands, the two strands, something like this. This is just like a sketch, all right? And then we have the strands, and then we have something like this. They are linked to each other, something like this. So looking at the DNA, according to Francis Crick's and James Watson, they actually describe the DNA strand as a twisted ladder or staircase that is capable of zipping apart. All right. Now, each strand of a DNA is made up of nucleotides. Each strand, meaning the right, the left, is made up of nucleotides. And these nucleotides, they are made up of one, we have the phosphate group. Then two, we have the nitrogenous bases. And three, we have the sugar unit, the sugar unit. All right. So these are the three parts of a nucleotide: phosphate group, nitrogenous bases, as well as sugar unit. Now, the sugar unit is usually in the form of ribose sugar. All right. And ribose sugar is a 5-carbon sugar. Then while nitrogenous bases, what are the nitrogenous bases? There are about 4 of them. Let me just quickly outline them. This one is still very important for those that are writing exam. All right. Now, looking at the nitrogenous bases, there are 4. And what are they? I go with this principle, the GCAT. That's according to Chagaf's rule. GCAT. That's Chagaf's rule in terms of how you pair them. Now, this stands for guanine. This stands for cytosine. Then this is adenine, while this guy is thymine. All right, thymine. So these are the 4 nitrogenous bases. Now, in terms of pairing them, the guanine always pairs with what? With cytosine. And the adenine always pairs with what? With thymine. All right, so this is how you pair them. They are complementary strands rather. So if you have G here should be C. If you have C here should be G. If you have A here should be T. Then if you have T here should be what? A. All right. So that's how you pair them. Okay. And what is connecting them together? The bond here that is connecting the 2 nitrogenous bases together is the H bond, which is hydrogen bond. All right. That's what links them up together. So these base pairs, they are actually grouped into 2 groups. Now we have the purines, and we have the pyrimidines. The purines and the pyrimidines, those that are writing exam, be very, very focused now. Now, the purines, what are the nitrogenous bases that you have under purines? You have adenine, and we have guanine. Guanine and adenine, they are the ones that are present in purines. Under the pyrimidines, what we have there is cytosine and thymine. So these are the 2 under pyrimidines. So please don't forget this. So this is very, very important that you know there are groups where they are grouped. Then if you talk about, there's something else that is called RNA. RNA. Never should you mistake RNA for DNA. RNA stands for ribose nucleic acid. All right. Now, this RNA is found in the nucleus, talking about the nucleoplasm, the environment of the nucleus. All right. While DNA is found in the gene. You get the point now. So the thing is this RNA, it has about 3 types. We have the mRNA, we have the tRNA, and we have the rRNA. All these, they stand for something. This is messenger RNA, this is transfer RNA, and this is ribosomal RNA. These are the 3 types of RNA. Now, basically, during enzyme synthesis or protein synthesis, remember I drew the cell and then I said this was the nucleus. Within this thread-like structures, that's where you have the gene as well as DNA. So let's just say the DNA is here. Now, this DNA is small, but then again, Within the environment of the nucleus, we have pore-like structures that are called nucleolus. It is the nucleolus that synthesizes RNA. So it means it is the nucleolus that produces the RNA. All right. So now the RNA is what gets information, because now the DNA cannot actually move out. It cannot move out of the nucleus because of the size. The size is too big. So since it cannot move out of the nucleus, it makes use of the RNA that is smaller, because the RNA is just a single-stranded molecule. And as a matter of fact, what distinguishes RNA from DNA? Let me just quickly outline it. What distinguishes RNA from DNA is that, remember I said that the DNA is made up of these nitrogenous bases, but the RNA is made up of these same nitrogenous bases: guanine, cytosine, adenine, but in place of thymine, is now replaced with uracil. So in RNA, these are the nitrogenous bases that make up RNA. All right. So it's different from the ones that make up DNA. The only difference is that in place of thymine, this now has the replacement of uracil. So uracil replaces the thymine there. Don't forget that. And RNA is single-stranded, meaning it's not double-stranded. It's just single-stranded. All right. So you don't have anything like pairing there. You don't pair in RNA. So during the coding of protein, or the synthesis of protein, or synthesis of protein, now what happens is this: the DNA will transfer the information by a process known as what? Transcription. Transcription. So transcription, it takes the information to RNA. So RNA picks up the information and takes it to the ribosomes present in the cytoplasm of the cell by translation. All right, translation. So basically this is just the process of protein synthesis or enzyme synthesis, from transcription to translation, and from translation to the production of protein. All right, that's called the central dogma of life. So that's that about that. We have seen the RNA, the DNA, the main things about them. Then let's quickly touch some aspects of this genetics, aspects like the Mendelian principle, Gregor Mendel. Now you are asked a question in JAMB, that who is the father? The father of genetics. The father of genetics is Gregor Mendel. That's the father of genetics. And he was able to postulate two laws. Now we have the Mendel's first law, the Mendel's first law of genetics, and we have the Mendel's second law of genetics. Mendel's first law and Mendel's second law of genetics. I'm going to be dropping something that will be of help to you now. Please be very focused. Now, Mendel's first law is also called the law of segregation. Don't forget that. The law of segregation. Then Mendel's second law is also called the law of independent assortment. Independent assortment. The law of segregation as well as the law of independent assortment. What does the first law talk about? There are so many things ascribed to the first law, but let me just give you the summary. What it simply states is that of the two genes inherited by an offspring from both parents, only one is inherited per parent. What does that mean? It means of the genes that are inherited from my father and from my mother, I inherited one from my father and I inherited the other from my mother. So that's what the segregation really entails. And the segregation usually explains monohybrid inheritance. The word mono means one. So when I say monohybrid inheritance, I'm talking about inheritance of just a particular one gene or character. You get the point now? Then there's also one called the hybrid. The one that talks about the hybrid is the second law, the hybrid inheritance. That one talks about the inheritance of two characters, two different characters. It could be different, or even they're always different. You get the point now? So two different characters is to dihybrid. Then while one is to what? Monohybrid. So when we talk about monohybrid inheritance, we are referring to inheritance coming from the sickle cell. Sickle cell anemia is one of them. That's under the monohybrid inheritance. All right. So we have the sickle cell anemia. Then we also have albinism. Yeah, albinism. So when we say sickle cell anemia, what are we referring to? The kind of illness that is caused when there is abnormal shape of red blood cells. Normally, the red blood cells ought to be biconcave in shape, but when the shape of now the red blood cells is kind of sickle shape, it means that the surface area has been reduced. And if the surface area is reduced, then of course that can result in what? Inability of the hemoglobin that is resided there to transport oxygen properly. And that can result in deficiency in oxygen in so many parts of the body. Deficiency in oxygen of the brain can result in paralysis, and in the muscles can result in rheumatism, then even in the abdominal pains, talking about deficiency of oxygen around the intestines, the stomach regions and all that. You get. So that's for sickle cell anemia. All right. Then albinism. Albinism is when a particular enzyme fails to produce tyrosine. Tyrosine. Now, the enzyme is called tyrosinase. Tyrosinase. So it means that if the pigment of the skin fails to produce this enzyme, then this enzyme will not be there to produce what? Tyrosine. This tyrosine is actually what produces melanin. And if you have watched my video lesson on the skin in this channel, then you should know that melanin is a dark pigment that is found in the mammalian layer of the skin, and it regulates the skin tone. So if there is no production of this, there will be no production of this, and there will be absence of melanin. And when there is absence of melanin, it results in what? Albinism. One suffering from albinism is said to be called an albino. So these two, they are monohybrid inheritance. Then we still have dihybrid. Dihybrid is a situation whereby you are having a pair of contrasting characters. For example, let's say you have rounded coated seeds that are wrinkled. Sorry, not wrinkled, they are green. Could be having yellow seeds that are rounded. Yellow seeds that are rounded, and you could be having um, green seeds. Green seeds. This one is capital letter. Green seeds that are wrinkled. You get the point now. So in this case, how many characters are expressive here? There are two. So we have yellow as color, then rounded as shape. And why this one is what? White. Sorry, wrinkled. This is wrinkled, while this one is green. All right. So you're having two of them that are being expressive. So that's under the hybrid inheritance. You get the point. Then as for the Mendel's second law, which is the law of independent assortment, what it means is that random genes, they tend to assort themselves independently. So it means that each pair of allelic gene may combine randomly with other pairs of allelic gene. So they are not dependent on any of the genes, they just combine randomly. So that's what the dihybrid inheritance talks about. But the basic thing that you need to take note of, because I don't want to go into deep details on this particular aspect, basic things that you need to take note of is that there are other names. Mendel's first law, law of segregation. Mendel's second law, law of independent assortment. All right. So these are the things that you must take note of. So that's that about that of genetics. But meanwhile, sometimes they might tell you to, especially the Mendelian cross, they might tell you to calculate the probability of having a child to be short or the probability of having a child to be tall, stuff like that. Now, let's just say a man. Let me give you possible questions that he might ask on this. A man gets married to a woman, and this man is homozygous tall. Remember, TT. Then why the woman is homozygous short. Now, if you do the Mendelian cross on this is TT. All of them will come as what? As heterozygous tall. If you check, remember that tallness is always dominant over shortness. So in this case, all the offsprings here, they are what? Heterozygous tall. So in this case, I can as well say that there is no homozygous short amongst the offsprings. But what about when you are given this, that a man that is heterozygous tall, a man that is heterozygous tall, and a woman that is also heterozygous tall. So what's the probability of you having the children, one of the children to be homozygous short? What's the probability? Let's confirm. Now for the Mendelian cross, we have TT, then we have TT. You see how I'm connecting them? TT, and we have TT. Check. This is homozygous tall, heterozygous tall, heterozygous tall, homozygous short. So what's the probability of having a child to be short? Is this guy, which is 1 over this 4. Probability is number of required outcomes over number of possible outcomes. So this is the only short. So out of 4 children, you'll be having 1 over what? 4. Then that is the probability of having a short child, 1 over 4. What about the probability of having an homozygous tall child? It's still 1 over 4. But what is the probability of having heterozygous tall child? Is what? 2. 2 over what? Over 1, 2, 3, 4. 4. So when you reduce this, it becomes what? Still 1 over 2. Alright? So that's how you go about it. That's how you calculate probability. Then, what about if they tell you the percentage? All you simply do, if you understand how to get probability, is just multiply by 100. So imagine they said the percentage of having a short child from this expression is 1 over 4 times 100. And when you do that, 4 into 100 is what? 25. So the percentage of having a short child is 25%. That's how you go about it. Let me drop one. Let me drop one. This one is very, very common, that they always advise um AS. AS. They will say AS cannot marry. The reason is because now if a man is AS and a woman is AS, when you carry out the cross, this is AA, this is AS, this is AS, and this is SS. So check. The probability of having a sickler amongst the offsprings is just this one. So that should be 1 over how many of them now? The outcomes 4. So if it's 1 over 4 as the probability, then what is the percentage? Very easy. Multiply by 100. That should give us what? 25%. But do you know what? This is just a normal theory explanation, meaning this is not practical. So some couples might think that, okay, now if we give birth to the first 3 children, we can stop there, and then the last will not give birth to the last. No. The thing is this is probabilistic, meaning the good gene has an equal tendency as the bad gene in crossbreeding. And what does that mean? It means the tendency of a good gene meeting with the good gene is still equal to the tendency of a bad gene meeting with a bad gene. So it means that the couple might just be unlucky that the only genes that are always meeting are the bad genes, and that can result in all of the children coming out as SS. So this is just like the explanation to how you can calculate those genotype in terms of probability and percentage. Then in this case, which one do you think will be phenotypically okay or normal? Is actually what these 3. So the phenotype for the ones that will be okay or normal is 3:1, because these ones will not be really having issues. It's only these that will be having issues. So that's how it works for phenotype as well. All right. Then what about maybe they tell you express the genotype in terms of ratio. This one is phenotype, meaning these 3 are okay, while this one is having issue. That's for phenotype. What you can see, these 3 children, they are okay. This one is not okay. That's phenotype. But for genotype, the ratio of the genotype, that genotypic ratio here, is this is 1. These 2 they are the same, so it's 1:2. Then this one is different, so it's 1. So this is the genotypic ratio of this expression, while this is the phenotypic ratio of this expression. All right, so take note of that. Then let's quickly look at variation. Variation is a part of this topic that poses a threat to so many students. So many students dey find it difficult to understand. So let's see what variation entails. I will just give you the basic thing about variation. When we say variation, variation like I said refers to differences in a group of organisms of the same species. The differences you can find, like what I meant by that is every member of a given family, they are not alike in every aspect, like they are not in resemblance in every aspect. Now your brother, your sister, they are different in terms of their facial expression, though they are from same family but they are different. You get the point? So that's what variation entails. Now variation can come as a result of mutation. That's why when you hear of mutation, mutation talks about changes that could happen either in the gene or in the chromosome. That's why when you hear gene mutation, is the changes that occur in the gene. Or when you hear of chromosomal mutation, you also know is changes that occur in the chromosomes. All right. So what are the two types of variation? I don't want to waste much time on this. We have the morphological variation, and we have the physiological variation. Morphological variation, physiological variation. All right. When we talk about morphological variation, we are simply referring to body forms and structure. Forms and structure. I just want you guys to understand this thing very well. That's why I'm summarizing this. Forms and structure. Then when we talk about physiological variation, we are referring to behavior, differences in the behavior of organisms as well as function. So that first talks about differences in the body forms and structure, while this one talks about differences in the behavior and function. Of course, in our world today, we have people that have different body structures and body forms, and we still have people that have different behaviors as well as different functions. All right, so let's concentrate on this guy, this morphological variation. What are the different types or examples of morphological variation? We have color, we have size, we have height, we have shape, and we have fingerprint. Fingerprint. All right. Then looking at behavior, we have blood grouping. We have blood grouping. We have tongue rolling. Tongue rolling. We have ability to taste. Ability to taste. PTC. That's phenylthiocarbamide chemical. All right. So these are actually physiological. Then there is something that you need to take note of. Morphological variation gives rise to continuous. Always remember this. Continuous variation. Then physiological variation gives rise to discontinuous. That's why in most texts they will just say that physiological variation is also called discontinuous variation, while morphological variation is also called continuous variation. So these ones, they are self-explanatory. Now for colours, we have different kinds of colours, and the colours can come in the form of eye colour, skin colour. When we talk about eye colours, we are having often brown eye colours as well as blue eye colours. The in-depth of that is, now if the melanin concentration in the iris of the eyes gives rise to colours of the eyes. So those that have a high concentration of melanin usually have blue eyes. So when there is low concentration or absence of melanin, it can result in blue eyes. All right, the first one is brown while the other is blue. When there is high concentration is brown, when there is low concentration is blue. So this size, some persons are literally fat while some are slim. That's size. Then height, some are tall, some are short. Shape, some have different body shapes. You understand? Then fingerprints. Now, when we say fingerprints, we are referring to those impressions on the epidermis of our palms, our palms. Now, if you check, even our toes as well, you check these impressions, they are different. No two individuals has exactly the same fingerprint, even up to the extent of checking identical twins. Identical twins, they don't have exactly the same fingerprint. They have different fingerprints. And fingerprints are of different types. And what are the different types of fingerprints? Now, we have the arc, we have the loop, we have the wall, and we have the compound, or you can go with composite. So compound or composite. So these are the types of fingerprint: arc, loop, all or wall, and compound. All right. So those are for fingerprint. Then looking at these ones, blood grouping. Now for blood grouping, we are having about 4 types of blood group that's coming from the blood group ABO system that was discovered by Dr. Landsteiner. I think that was in the year 1902. So that blood group ABO. Now we have blood group A, we have blood group B, we have blood group AB, as well as blood group O. So these are the 4 types of blood group. Of course, the one that is called universal donor is O. Universal donor is O. Then while universal recipient is AB. What does that mean? It means that this universal recipient can receive blood from all types of blood groups during blood transfusion. Then while O can only receive from O, but it can donate to all. All right. And then you know that when you check your blood group, is that you are seeing A positive or A negative, this one B positive or B negative, this AB positive, AB negative, O positive, O negative. Now, where those positive and negatives are coming from is as a result of resource factor. Now, the rhesus factor was first discovered in an organism called the rhesus monkey. So it was later, after about, I think, 38 years, when the blood group ABO system was discovered in man by Dr. Landsteiner. In combination with Dr. Levy, they were able to discover the rhesus factor or antigen in man, and that was 1940. So it was around 1940 that they discovered that. That's a rhesus factor. Rhesus factor. Rhesus factor. So there are some persons that are rhesus positive, while some they are rhesus negative. So those persons that have rhesus antigen on their red blood cells, they are rhesus positive, while those that lack it, they are rhesus negative. So these positives are those that have it, while the negatives are those that don't have it. All right. So that blood grouping, one thing that you need to know is this: antigens, antibodies are of relevance here. Antigens are found on the surface of red blood cells. Antibodies are found on the plasma. Remember, if you have watched my video lesson on the transport system in this channel, then you get to know that the blood has plasma and blood cells. The plasma is the liquid portion of the blood. All right. And looking at the antigens, antibodies, like I said, antigens are present on red blood cells, antibodies on plasma. So these blood groups, blood group A has antigen A and antibody B. Blood group B, antigen B, antibody A. Blood group AB, it has antigen A and antigen B, but it does not have antibody. So it means if you are asked in the exam that what blood group doesn't have antibodies, is AB, no antibodies. That's the reason why AB is universal recipient. I will explain that. But meanwhile, for blood group O, is the only blood group that does not have antigens. That's the reason why it's called universal donor. Let me now explain something. During blood transfusion, you must have the donor and the recipient. The person that needs blood and the person that is giving out blood. The person that needs blood is the recipient. The person that is giving out blood is the donor. Now, what is considered of the donor's blood is what? Is the antigen. Then what is considered of the recipient's blood is the antibody. So if they do not agree, if the antigen of the donor does not agree with the antibody of the recipient, it results in a condition which is called agglutination. Agglutination, or you can call it what? Blood clumping. And that can ultimately result in death. Yes. So the thing is this: if there is no agreement, then it can result in that. So it means blood group O can donate blood to all blood groups. Why? There is no antigen to react with the antibody of the recipient. That's the reason why it's universal donor. Remember I said what is considered of the donor's blood is the antigen. Then what is considered of the recipient's blood is what? Antibody. So that's why you see this guy being universal recipient means that he does not have any antibody. So there will be nothing to react with the incoming antigen. You understand? So that's the reason why. So please take note of that. Take note of that. Okay. So let's look at tongue rolling. This tongue rolling, there are some persons when they stick out their tongue, they cannot make a U shape of the tongue. While there are some, they can make U shape of the tongue. So those that can make U shape of tongue, they can roll their tongue when stick out. They are called rollers, tongue rollers. The while those that cannot, they are non-rollers. So if I this is actually I'm a roller. So then why some they can't? So they are non-rollers. So that's for tongue rolling. Then ability to taste PTC. PTC I said is phenethylcarbamide. So this is a chemical. Now some persons they are tasters while there are some that are non-tasters. Now the tasters will tell you that this chemical is bitter in taste, while the non-tasters will say it is tasteless. So basically, just take note of their respective examples. Then also, the application of variation is still very important. Yes. When do we apply variation? Where is it applied? Variation is applied in different areas. Now, the first is for crime detection. You can use it for crime detection. You can use it in the determination of paternity, determination of paternity, as well as marriage counseling, all right. Now why variation has these different applications is because now, if you check crime detection, those civilized nations of the world, now they can use the fingerprints that are left by criminals to actually get the criminal. Yes, all those opening of door, as you open door, your fingerprint is registered on the door. They can extract it and they use it to get the criminal of that particular incident. All right. Then for determination of paternity, when I say paternity determination, what does that mean? It means to determine if truly a man is the father of a child. All right. And you can use the variation, the application of variation to doing that, especially on DNA sequencing. DNA sequencing. You can use DNA test. Yes, you must have heard of DNA test. That's a situation whereby you can use any part of the child's body to and pick from the father's body. Then you combine to see whether it agrees. All right. That's DNA test. Then we also have marriage counseling. Now, marriage counseling, of course, it is due to the application of variation that you can actually advise on how to get married. Now, in terms of genotype. Now, there are some persons that may not want to go with the advice, but it's advisable you go with it. The reason is because AA can get married to all genotypes, but I once explained something with regards to AS, that AS cannot get married to AS. The reason is because there's every tendency of a child coming out, possibly as SS. So that's why they often advise not to get married. All right. So it's with my hope that I've been able to touch things that can be of relevance to those of you that are preparing to write in exam in the forthcoming JAM or in the forthcoming WAEC exam. I'm wishing you guys all the best. You are yet to be a subscriber or a follower of this channel. Do well to follow. As much videos are yet to come, invite your friends too so as for them to benefit. All right, because these video lessons that I'm dropping are video lessons that will be of help to you. I wish you guys all the best.